Okay, so I've got 3 papers based on the oral metagenome in both health and disease. So these 3 studies show what's going on in the oral cavity. The first paper is by Belda. This paper is done in Spain, and what they've done is they've looked at pyrosequencing of 8 people, or 8 samples. In this paper, they're saying that the cavities are not dominated by S mutants. Next paper, the personal oral microbiome obscures the effects of periodontal disease. This paper was done by a group of people. You can see San Diego, Italy, Los Angeles, Illinois at Argonne Labs. So lots of people on this paper. And the sample size here, I'm trying to recall, I think it's much larger. Yep, here it is, 36 people. So they're looking at 36 people for the Schwartzberg paper. And the last paper here that I picked was a study from a lot of people out of Boston. Bo Liu is the author of this paper, and they're looking at the microbiome of 5 people. Now I'm trying to find it in the abstract. I don't see it there, but if you look at the data, right there, person number 1, 2, 3, 4, and 5. So let's take a look at the Belda paper. What they're doing is coming up with the oral bacterial diversity, and I need my glasses to see.
I know.
Okay, so let's take a look at the model at the bottom. You've got the range of tooth health, where dark blue is not very good health, light is good health. You can see how it clusters. If this is all healthy, this is what's in the healthy tooth, and this is what's in the not healthy tooth. The patient's NOCA001 means no cavity, person 1, no cavity, person 2, and then CA1 is someone with a cavity, and so on.
So how come the person with no cavity has the darkest, like,
in the top?
Yeah.
Okay, so let's take a look at the paper. Make sure I get that right. Belda. Did I read the
Relative frequency? Fine. Go up. That means the frequency. It's just the frequency of bacilli.
Oh.
It's not the cavity causing. Go to the other side. Yeah, here, in the orange one.
Rho Z score.
Yeah.
So they're not coming up with.
No, they're just telling the abundance of these particular species over there. It's not like.
Oh, and here's what they've said. So these are the healthy teeth.
So
these are the unhealthy teeth.
Healthiest teeth, the abundance is of these gram, whatever, second, and bacilli, and clostridia, bacteria, actinobacteria. These are the ones that have been associated with. Yeah, which we would expect. The spirochetes and the ones below are the ones which are associated with the.
I was expecting a grouping with bad health. So
No,
you can see Clostridia,
just
Bacteroidea, maybe these ones with
No.
more cavities.
No, the dark one you're seeing in the non-cavity one as well. I think those
Oh,
are the
but
ones
here,
that they are.
see this little area right there?
Yeah.
Might be a little different than this area right there.
Yeah. True, but the ones with
And this
active-
area right here, this is no cavities versus these guys with cavities. So maybe these two groups are not in cavity regions.
Yeah, relative abundance it is. So yeah, okay. And what did they say about the mutants? Just can you identify it again?
Go back. Streptococcus?
Yeah.
Where is Streptococcus? Bacilli, right?
Yeah.
They're at the top.
Yeah.
Most abundant.
Yeah, but this
Streptococcus are in everything.
everything else as well, all other
Everywhere, yeah.
acid-producing bugs. And they are abundant in the non-ones as well.
Okay, and in the right-hand side, they're looking at the different,
Techniques.
different categories.
What is
So
RNA processing over here?
how many genes were found that are RNA processing genes? This many. How many genes were chromatin related? This many.
Oh, so functions, cell functions.
Yes, this is a functional breakup.
So secondary metabolism and recombination repair and all those things are really high in these most abundant bacteria.
They're trying to make a comparison with maybe the Clostridia. If this band here is darker than up in,
Yeah,
so
exactly.
health might, you might be able to figure out how to make something healthy.
That's interesting.
I like that idea.
Yeah.
I think that's a really strong idea. Actually, you know what we could do is, because this paper is kind of hard to read in a small format, we'll just go through the images. There's figure 1, here's figure 2. That way I can make the whole figure here. Okay, so are you familiar with the way they do metagenomic principal component analysis?
I'm not sure.
Okay, so there's usually a two-dimensional graph, and the two-dimensional graph locates a variety of I'm trying to remember. It's not phylum. It's one up from phylum, right? It is? You're agreeing?
No, I think it's
so like something like Corneobacterium's up here, Campylobacter, Actinomyces. There would be a spectrum on the left-hand side, -1 being healthy, all the way to +1 unhealthy. And so when you take a look at the bacteria in a two-dimensional way from healthy to not healthy, there's also a second component that you can analyze this by. I'm not too sure what that axis is. They don't mention it in the paper, but you can see they're also breaking up the bacteria this way as well.
Show me the paper.
I don't have it printed, sorry. I know. We could look. This is table 2, right?
No, I just want to see what is CA2. Go down.
CA2, it's the correspondence analysis.
No, that is CA generally, right? What is CA2? What is the y-axis?
Well, there's the corresponding analysis, axis 1
yeah.
and axis 2.
Yes, that's what I'm asking. What is this axis 2 signifying?
I can tell what axis 1 is by in the paper they said, but axis 2 I didn't find it. Let's see, here's figure 1.
They have CA0.
Here's figure 2. Okay, so it's going to be in here someplace.
Do control find
Corresponding analysis between metagenomes
and taxonomic
assassination by 16S showed samples with poor oral health clustered together.
Yeah, so it's fine. So one is, I think, 16S and metagenomics.
It could be, right? So,
But
like
that
I
was
was
for
saying,
figure 2.
this is figure 2, yeah.
Yeah. Yeah, so that's what they're doing. 16S
So,
versus
CA2 would be relatedness between the bacteria.
or 16S,
16S,
right?
which is
And
16S.
the
And
lower
the
one is the
tooth health.
and yeah.
So they're breaking up all of the bacteria, the 2 dimensions that way. Let's go back to PowerPoint. That's that slide. Here's the functional profile, and I was just saying that you can break up the tooth health now by functional profile. Let's see, here's a healthy, the N2 are healthy teeth. Are these 2 columns different than unhealthy teeth? Can you find a visually obvious difference? And you can see here these genes being overrepresented, whereas in the unhealthy, they're underrepresented. Next slide. Bacterial strains with antagonistic effects against karyogenic bacteria. So they're looking at putting different bacteria together. They found S mutans in the healthy tooth, no cavities.
Yeah, which we kind of know that it is present. It's not karyogenic. It's
Right.
facultative pathogen, right? So
And here it is in
really
in a
bad tooth health.
cavitated tooth, yeah.
What is the thing that it's showing? The colors. What is that signifying?
So let's take a look. That's why I have the other paper open. So
Go up. Yeah, that one. See the different colors. So you see a difference between what you're seeing.
CA04 and no cavity 01.
Yeah.
This is
Read what is written under this.
they have A, B, C, and D, so 4 stages. So they're looking at the antagonistic effect of these bacteria, and these 4 pictures are metagenomic recruitment plots. Have you ever heard of these before?
No.
Me either.
But what they are saying is which are low frequency
Low
individuals
frequency.
with caries but are among the most common in caries-free subjects. And if ever you see the plots, it's whatever the colors are signifying. It is way more in the ones in caries and way less in the ones in non-cavity ones.
Yep.
So, I don't know. The sanguineous is, I think, more as a positive one, so it's more on the other side.
Yeah.
Right?
It's in no cavities.
Yeah, so if ever we
Whereas-
see that way, it is kind of showing that mutants, whatever that color scheme is signifying, it's more in the cavity one, and sanguineous is more in the non-cavity one, which we are aware of. So I don't know how they're saying the other way around.
They also isolated
Mm-hmm.
a new species, 7747.
What is that? Some streptococci?
It's, see how it's related. It's not Peroris, it's not Infantitis.
It's a family over there.
Yeah. They're calling that an inhibitory strain because it inhibits other Streptococcus. Okay, that's that paper. Schwartzberg. So this study is the one with 70 people. They perform something called a Procrustes analysis. And what they do is they take these people's sample and they look at before and after treatment. And then what they do is they visualize what the community is like before and after for a number of well, so same sort of thing. So you have PC1, principal component analysis, likely to be health, PC2 likely to be 16S, genetic distance, and a third value. I don't know how they're getting the third value, but they're putting it into three-dimensional space. Oh, there's another number, principal patient improvement or decline based on changes in observed pocket depth. So that could be your third axis. Next, they do a number of curves, and they're showing the trends of the samples. So if you sample at different pocket depths, Fusobacterium, you notice, increases the deeper you go, which is kind of expected for that spirochete. Prevotella, the deeper, oh, this is a different one. This is
percentage streptococci to percentage Prevotella.
Right.
So as the Prevotella increases, streptococci decreases.
Right.
Which is what we expect as well, right?
Yeah.
So that's fine.
And it's the same thing with Streptococcus and Loshi. They also perform refraction curves to show that they've saturated the metagenome of these people. So if you have seen a rarefaction curve before, if it's going up, then you know you haven't sampled enough. But if it reaches a saturation point, you know that you've sampled enough. But they never seem to go level at the top. They're always continuing up. Yeah. Meaning you needed more money to do the study, to do more samples. Of course. Next. Now this is kind of, really messy. Trends of bacterial genera associated with health or disease. So here's the Fusobacterium in letter A. They're looking at people who worsened after treatment or improved after treatment. So if you take a look at the lines, for the people who have improved, what happens to the line? For the people who get worse, what happens to the line? So Fusobacterium is increased here, and if they improve, it's decreased, which kind of makes sense.
It's like, again, the same thing they're showing in a different graph.
It's a very complicated graph, but it can be understood if you play with it and look at it long enough. So Prevotella, a bad bacteria, let's see, it goes down if you get, but it goes down even further if it improves. So it goes down if you don't get better, if you get worse.
No, but it's going down in some and it's going up in some.
True. The overall, though, is this number. Streptococcus, if you get worse, it goes down a little bit. If you improve, it doesn't change. And Villanella, if you get worse, it goes down a little tiny bit. And if it improves, it goes up. I think Villanella is a good one.
Well, right? No, obviously is a good one, because it neutralizes whatever, right? So.
Yeah. Also, the number of people that they're treating or examining here for this study is less.
So that's why they have this problem.
Yeah. Not a lot of data.
Yeah.
Now, if you take a look at all of the Prevotella and try and figure out where the problems are, look at this one. This number stands out from all the others, where it's really, really much different. So the changes in the proportions are species dependent. That's what they're saying.
Yeah.
Here's P. denticola. It's
P denticola?
P. Prevotella denticola.
Okay.
I
They
said
changed the name?
I didn't think so, but there might be a T denticola, or a P denticola as well as a T denticola.
Because I
I
heard
always know it as T.
I know.
True.
They might have changed the name as well. I don't know. Because they
We could look it up after.
Yeah.
So that's your cladogram of, okay, the cladogram of Prevotella, and here's the cladogram of Fusobacterium. And the Fusobacterium isn't as nicely laid out, it's just the orthological taxonomic units that they're showing here. They're saying that Fusobacterium is very diverse. That's it. Last paper, Lou, and Lou did this deep sequencing project. And this is the Harvard paper. Well, some people from Harvard, some people from Maryland, but a lot of these are from some association with Boston. Okay, here's the first table, and as you can see, they looked at 5 people and read the shotgun reads from those 5 different people. If you just go along here, who's the subject? They just number it. This is subject number 1, tooth number 14. What is the state of the tooth? Advanced disease here. Down here it's a healthy tooth. How much DNA did they get for reading? This tooth had 51,000 reads, which isn't really a lot.
Well, relatively abundant, right? Because they are able to. I think it's enough because it's really high to start off for the reading, right? So that means.
Well, this number is much better. That you get 9 million shotgun reads from that. The problem is there's a lot of human DNA in some of these samples. And what is the dominant gene in the person who's sick? It's Prevotella. Prevotella, sick. Streptococcus is a healthy person, and Actinomyces is another healthy person. Next paper, or next table, the statistics from the shotgun reads. This is just all the statistics that come out of it based on the 3 ways of reading their data. So they do something called SOAP de novo, another comparative, another where they take SOAP de novo, add the comparative test, and by doing that bioinformatics they get more data from all of their samples. So they did 2 with periodontitis and 2 with health. As you can see from back here, for some reason,
they had nothing.
Nothing came from this healthy individual. So the student handled the DNA badly. I don't know.
They couldn't isolate.
Couldn't get it proper to work for them. Yeah. Okay. So they're also doing an assembly of TM7A. TM7A is one unculturable bacteria that they pulled out of this, that they analyzed to a larger degree.
Why do they call it TM7? Because it's from the TM region?
Could be. I don't remember seeing why. Next, they do the abundance. And so H21, H11, if it starts with an H, those are your healthy people. P, those are the people with,
So in healthy, they have a high abundance of actinomyces.
actinomyces,
Second last, white.
high, yeah. And streptococcus, which is
Okay because red
almost
is
what
low,
we saw in that last paper.
Yeah, but I'm surprised that they also have actinomyces.
And look at P1, P12, P13. Streptococcus and actinomyces are
Are low.
almost
Yeah,
non-existent.
they have an issue with their sequencing.
And Fusobacterium and Prevotella, very high.
Relatively high.
Yeah, so I think the red complex that Bailey talked about sometimes includes Prevotella and Fusobacterium. Now, I know by Cox postulates they can't come up with that one bacteria causes it. It's always a community of bacteria.
Yeah.
In this paper, what they also do is they say, okay, well, let's look at the genes and where do we see clustering? An interesting cluster is in fatty acid, either metabolism or biosynthesis.
Mm-hmm.
If it's blue, it would seem to be enriched in healthy samples. So healthy people have increased fatty acid biosynthesis. Sick people had increased fatty acid metabolism genes.
This is membrane alterations, right?
Yeah.
It's the metabolic pathway of the bacteria or metabolic pathway of the patient.
Bacteria.
So that fatty acid metabolism would mean like the membrane alterations of this, right?
I thought this was the bacteria, but I should look at it again.
No, I think it is bacteria because.
Yeah.
Yeah, I would guess it is bacteria.
Okay, the confusing curves from the metabolic shows, I call them confusing, but they're pretty simple. There's this one bar graph that's called the Shannon diversity. Now, as you can see, one group is higher than the other group. If you have a diseased sample, your community is more diverse. That's what the Shannon Diversity Index says.
I don't really think so. I somehow feel the comparative.
It's the other way?
No, I feel that they cannot claim that it is more diverse. I feel it can be either way because see the comparison between blue and pink for the first one and blue and pink for the second one. It's the diversity difference is almost the same. The control genus and the case. The control phylum and the control whatever. I somehow feel that. And look at the standard deviation.
Well, yeah, if you take a look at these error bars, they're all both crossed.
So
you can't say they're not really different if you're going by that.
Yeah.
Maybe they did a different type of analysis, and based on that analysis.
And I somehow feel statisticians can come up with
yes,
very significant
they can figure that out nicely, can't they? Okay, the next picture, letter B, here's another principal component analysis. So they have the 16S on one side and
whole genome sequencing data. WGS.
Yeah. And what they're saying here is that when you do perform this analysis for the taxonomy, your disease clusters in the bottom left corner.
Yeah.
So the disease are these red circles and red squares, and the healthy ones, they're all over the place. Nice analysis, right? Letter C. Okay, again, you can see the unhealthy samples cluster in the bottom left corner doing this enzyme analysis.
What enzyme is that?
Well, like they did here.
Okay, so some enzyme.
Right. Different enzymes.
Okay.
So they're showing that the different enzymes
That
may
yeah.
be associated with disease or health.
Yeah.
Now, the picture I have a problem with is this Kmer frequency. They say relative frequencies of tetramers. And what is a tetramer? A 4 base pair motif. So they're saying that, okay, well, let's take a look at the tetramers. If you have a disease, the relative frequency is fairly linear. But if you have no disease or you're healthy, these tetramers are not linear.
So they are saying the disease samples are more similar to each other.
Yep.
As compared to controls.
Right.
What is the relative frequency over here? What relative frequency are they talking about? What is Kmer? How do they do it?
The 4 base pair motifs.
What is this 4 base pair motif?
Let's take a look, see if we can find this in the paper. So this is figure 3. Let's read the whole screen. This was letter D. There it is. Okay. So comparison of relative frequency of tetramers in metagenomic reads across disease cases.
No, that's the same thing that you have written. Can
Right.
you read it in the text? Like
sure. Let's find figure 3. Figure 3. Okay, so at the bottom here, it'll say
no, it's not
the disease samples harbor more diverse microbial community.
That's
okay, that's 3A,
yeah.
3B,
There.
3C. We want 3D. Where's 3D?
I know, wasn't it there up on me?
I didn't see it. I thought I saw 3A, B, and C.
3A, B, C.
Let's go look.
Yeah,
3A, B, C. No, figure 3D. Well, maybe we could look at tetramer. There, figure 3D. Okay.
As well as pairwise comparison of individuals and disease sample based on tetramer, subsequence of length 4 all indicate that disease samples are more similar to each other. I think it would be there in methodology as well, the tetramer. 10 matches, right? So check the other match.
I don't know how.
There is no find next.
Not in this program.
Okay.
There is just scrolling through everything.
Okay,
There's
3,
2
4,
more.
Keep going. There.
Calculating tetramer frequencies. Okay.
So there are phylogenetic signals. For each patient, we counted occurrence of each sequence motive of length of 4 for all the pre-processed sequence reads. Specific motive
So
divided by total number of
CTAG,
motives of length 4.
TAG, T.
Using these normalized values, we can visualize the figures.
So they're saying.
But who determines these motives? Maybe they're known and
they're-
they're phylogenetic.
The motifs are just the 4 nucleotides.
Yeah, but so they are just coming up with random, because see, they have specific combinations. It's not like just any. So obviously it is predetermined, like how to do it, or there must be a reason. And that's why they have organized it in phylogenetic
They say here,
clustering.
tighter linear group of the disease samples suggests the community are composed of organisms less diverse
Mm.
compared to the healthy,
Yeah,
which has more.
Which is
Which is
just
good.
like different way of saying the same thing
Yep.
and complicating it. Wow, you did so much.
So here's their model. So in a healthy tooth, you have good fatty acid biosynthesis, nutrients go in, and you get a healthy tooth. With a diseased tooth, limited oxygen, and you get fatty acid degrading things.
Oh, so they're talking about the patient, they're not talking about bacteria there.
Oh, okay. Yep.
Oh, okay.
And they did comparative analysis of this one bacteria, Actinomyces naislandii. They used that as a reference genome for their one bacteria that they found. Okay. Thanks. I think that's it.